This week on the Galactosemia podcast, I talked to Dr. Judy Friedrich-Kyle, Dr. Shauna Rasmussen, and Madeline C. Miller, who all just released a paper of their research project where they studied if bone marrow transplantation could have an effect on restoring GALT enzyme activity in the cells of a rat with galactosemia. And as far as we know, it's the first time that anyone's ever studied bone marrow transplants and the effects that it can have on galactosemia. It's a really interesting study that they did, and in the end they did find that it resulted in GALT activity in red blood cells, but the GALT activity and galactose metabolites in both the liver and brain samples remained essentially unchanged. And we talk about why that means that they conclude it is therefore not an effective therapy for galactosemia. We also chat about how the idea was thought of for this study, what the hopes were, how they ran the study, and what they thought about the outcomes. As always, you can get early access to these episodes as well as listen to them completely ad-free on the galactosemiapodcast.com. Thank you to everyone that goes there to support the show. Without further ado, here's this week's episode on bone marrow transplantation.
The Galactosemia Podcast.
All right, welcome back to the Galactosemia Podcast. We are here today with a few guests, and I'm so excited. So thank you all for joining. With me today, I've got Dr. Judy Friedrich-Kyle, Shauna Rasmussen, and Madeline C. Miller. Is that correct? I got it all right? Okay. So Maddie and Shauna are part of, or were part of, the Friedrich-Kyle lab that Dr. Judy runs. And if you are a frequent listener of the show, you've probably heard Dr. Judy on a couple of times. This is her third time on the podcast. So Dr. Judy, thank you for coming back.
My pleasure.
Always great to have you on. Shauna and Maddie, I'd love to start with just a little bit of about you. Maddie, why don't we start with you? How did you get involved in the Friedrich-Kyle lab? Where do you find yourself now? And who are you?
Hi. Well, thank you very much, first of all. I joined the Friedrich-Kyle lab my sophomore year of college, and I want to say 2021. Was involved in various ongoing projects in the lab and graduated last May, 2 Mays ago. So college graduate now, and I am at the NIH currently doing their intramural postbaccalaureate research program.
Nice. And I feel like that's like a whole other podcast we could do.
That's a whole other tangent.
The fact that you're at the NIH during all of what's going on right now, we'll save that for another episode, maybe.
Yes, for sure.
Shauna, what about you?
All right, so I'm Dr. Shauna Rasmussen. I am an assistant academic research scientist in Judy's lab, and I've been with her for just over 8 years now, and I have been integral in developing the rat model and then pursuing the different experimental interventions that we've been looking at over the last couple of years. And essentially, my goal here is to continue that type of work over the next few years.
Love it. I mean, we've talked to Judy about the rat model in the very first episode, and so there's a lot of interesting stuff there. Hopefully we'll get to dive into some of that in a little bit. But I'm curious to know from each of you, and maybe Shauna, we'll start with you since you've been in this field for a little while now. What was your first exposure to galactosemia? Did you know about it before starting with Judy? And you know, where did you first come across galactosemia?
So I knew nothing about galactosemia when I joined Judy's lab. So I was finishing my PhD and was looking for a postdoc position. And my boss at the time had said, hey, there's a lab down in Atlanta that is looking for a quote unquote rat person, essentially someone that had a lot of experience working with rats over time. And I said, hey, I worked with rats for 5 years. I would love to come down there. And so I interviewed for the position and then was able to secure the postdoc. And pretty much right when I started is right when Judy kind of started working with her rats. And so I've been working with them day in and day out for the last 8 years. And it's been very interesting to learn about the rats and the patients at the same time, because I knew nothing about the patient side of this. So it's been really interesting to see the similarities and the differences that our animal model and our patients have in common.
Yeah, that's super interesting. So you were much less of a galactosemia person and more of a rat person to
Correct.
get involved.
Yes.
That's awesome, but it brings some expertise that sounds like it was much needed.
Oh, yeah.
And I'm sure get used a lot. Maddie, what about you? Where was your first exposure to galactosemia?
First exposure was also Judy's lab. It's a rare disease, so I hadn't heard about it before, but I was looking to join a research lab to get some practical experience with research during undergrad, and funny enough, one of Judy's other undergraduate employees, Snay, was my chemistry tutor. And so while he was helping me fail chemistry, he told me about Judy's lab and galactosemia, and I said, screw chemistry, we'll do this instead. And so they introduced me to the lab. Judy introduced me to galactosemia and gene therapy and translational research, and it's been love ever since.
Wow, that's it's always interesting to find out what people's entryway to galactosemia is, because it's such a rare thing, and my entryway is my son had it. Judy's entryway is that she's an expert in it now, and then like you guys being part of the lab is amazing. And Dr. Judy, before we jump into kind of the meat of this episode, you and I talked a few weeks ago, and you mentioned a challenge that you guys face, which is losing touch with some of the individuals and families who have joined studies of yours in the past and wanting to reconnect. I wanted to touch on that a little bit at the top of this episode because I think it's super important. Can we talk about that really quickly?
Sure, and thank you so much for asking about that, Ryan. So we have a human subjects study that is basically longitudinal, which means we want to follow people over time. So we ask for updates every 6 months for little children and annually after age 6. And it's observational, so we don't ask people to change what they're doing, just tell us about it. And I recently enrolled patient number 631. We started in 1992, and I had been at Emory for less than a year. And it's really been an amazing experience for me. One of the wonderful things about running a very long-term natural history study, I guess, is that I've gotten to watch kids grow up. I mean, there's a crayon drawing in our student office. It's behind Shauna's camera right now, so you can't see it, but it's a little crayon drawing, and it's by a little girl who was about 8 years old when she drew it, and she said something like, "You can have my blood anytime you want it," or something like that, which I was just like, oh my God, that little girl is now a school teacher. Okay. So anyway, one of the privileges and blessings of having a long-term study is that for people who, I'll say, stay in touch, we can really see what changes and what doesn't change over time. The challenge is that there are a fair number of people who were probably enrolled as children by their parents, and then as they became teenagers and they became young adults, they've kind of stopped responding to us. And sometimes like an email address changed and a phone number changed, and I honestly don't know how to reach them. And there are a lot of privacy rules that we have to abide by, so I can't just send you a list and say, hey, do you know these people? So I'm kind of in a little bit of a bind. The folks who were in the study as children long ago, who we have lost touch with, I would love to get back in touch with them, and I promise we would make it as easy, and it's free, and we even send you gift cards if you fill out your surveys, and no extra needle sticks. But it would really be valuable for us to be able to follow up with people who are now, 10 years after we last heard from them, because we collected a lot of information and information from some of the blood or saliva samples that we got when they were kids. And part of what we're looking for is predictive markers. And unless we know how they're doing now, we kind of can't use that information from when they were a child. So I'll say we're also always looking for new people. So if you think you might have been in our study years ago and you haven't heard from us recently, please reach out to me. Send me an email is absolutely the best way to reach me. And it's just jfridov as in Victor at Emory, EMORY.edu. And I promise I'll respond. And so if you think you might have been part of our study, please contact me. If you've never been in our study, we're always recruiting new people. And it doesn't matter what age you are, and it doesn't matter whether your outcome is good, bad, or somewhere in between. If you've got galactosemia, come on down. We want to work with you. Please join the team. So
Yeah.
thank you.
And I can speak to it as somebody whose son is in the study. It's not a lot. You're not requiring a lot or asking a lot. It's probably 30 minutes to an hour every 6 months that you need to sit down and fill out some surveys. They're pretty simple questions. Is your son or daughter able to do this yet? Are they doing that yet? Are they? And at least that's my experience with a 1-year-old. I'm sure the questions are different for a 8-year-old or a 15-year-old. But if you're listening to this, you're probably someone with galactosemia or somebody that has a son or daughter that has galactosemia. That's the overwhelming population of people who listen to this podcast. So that means you should probably be in touch with Dr. Judy if you're hearing these words.
Please.
Send an email over to Judy. And I'll put your email in the episode description as well.
Great,
So if anybody,
thank you.
all you got to do is tap on that email, send her an email, and even just say hello. Okay, so
Thank
let's
you.
jump into kind of the meat of this episode, because what spurred us talking was an email that Dr. Judy, you sent a few weeks ago about a study that was done where you guys looked at bone marrow transplant. And so I want to jump into that study and just kind of find out what you were looking to learn and where you were trying to go and what you hope to learn. So why don't we start high level and tell us about bone marrow transplantation and why it's even something that you guys wanted to study in galactosemia.
Sure. So bone marrow transplantation is just like what the name suggests. It is taking bone marrow from one source, usually a donor, and giving it to a recipient. And bone marrow transplant is a very well-established procedure. It is standard of care. For instance, if someone develops leukemia or another disease that impacts the blood, then blood, the cells that give rise to both red blood cells and white blood cells live in your bone marrow. And so that is an extremely important tissue. And it has what are called stem cells, some of which just give rise to more stem cells, and others of which start to differentiate and become red blood cells or white blood cells. And so if a person has a disease that affects their blood, like let's say leukemia, the treatment is typically they give them some type of medication or radiation to pretty much kill off their own bone marrow, which has become cancerous, and then they give them back donor marrow. And that donor marrow, assuming it is a good match, so you've probably heard, people are looking for a match so that it won't be rejected by the immune system. Then that bone marrow, that donor bone marrow, basically, sets up shop in the marrow of the person's bones and starts growing and making blood cells, and that's called engraftment. And so it is a very well-established procedure. Now, galactosemia, we don't usually think of it as a blood disease, and so you might say, well, then why the heck? This is a crazy idea. And really,
That's exactly what I said.
okay. Well, so let me explain. So it's not crazy. It was a little fringe, but not crazy. So some of the white blood cells that come out of the bone marrow are not going to live in the circulation forever. Some of them actually, they're called macrophages, and there's a whole bunch of them. And they can actually leave the blood vessels and enter tissues. And when they go into the brain, they become microglia. And when they go into the liver, they become Kupffer cells. So the bottom line is there are cells that are derived from bone marrow that are present in lots of different places in the body, not just in your circulation. And I thought, well, gee, what if we could use GALT positive donor cells, then we could, most of the person's body, most of their cells are still going to be missing GALT, but we could have the cells in their bloodstream and the cells that leave their bloodstream and go wiggle into the brain or the liver or wherever they go, we could have these GALT positive cells kind of in the neighborhood, you know. And I thought maybe having these GALT positive cells, some of them in circulation and some of them not in circulation, maybe they could metabolize the galactose or the metabolites that are otherwise building up and act kind of like a sink to get rid of the metabolites that otherwise build up. And so that was, that was kind of the simple-minded way that I was looking at it. There are examples of other metabolic diseases that have been cured by bone marrow transplant, but the ones I know of are what are called lysosomal storage diseases. They are a special case because the enzymes that are typically missing in those diseases are enzymes that work in a special compartment of the cell called the lysosome. And there is a mechanism that allows cells to basically share their lysosomal enzymes. It's a mannose-6-phosphate. It basically, there's like a little tag that is on, it's a little sugar tag that is on the surface of these enzymes, and there's a special receptor that can grab them like out of the extracellular space and take it into the cell and send it to the lysosome. So if you have a child whose body is not able to make the lysosomal enzyme that they need, and you give them bone marrow cells that can make that enzyme, those bone marrow cells can basically share that enzyme. They can spit it out, and the other cells in the body can take it up. So that, unfortunately, GALT is not a lysosomal enzyme. So it can't be shared through the mannose-6-phosphate route. We're trying to come up with some other ways that maybe it could be shared among cells, but I was just thinking that if we had some GALT positive cells in every neighborhood in the body, that they might do some good.
And it seems like a good hypothesis and a good question. Was bone marrow transplantation studied in galactosemia before this at all?
Not that I have ever heard of.
Okay, so this was kind of like probably the first venture into that. And
yeah.
So I'm wondering if you can clear something up for me, because I think it'll pay off when we get to the end of this or when we talk about this study a little bit more thoroughly. In my understanding, and correct me if I'm wrong, through learning a little bit about gene therapy and stuff like that, in someone with galactosemia, the GALT enzyme's not active, and it needs to be activated in the cells that are in 3 places, kind of as I understand it, which is like the tissues, the liver, and the brain. Is that kind of it?
I mean, the truth is, normally speaking, the GALT enzyme is in every cell in your body.
Okay.
And we don't really know if having it in some cells and not in other cells would be good enough.
Okay.
So, and that's an open question. So we don't know. It's a pretty good guess that the brain is involved, and it's a pretty good guess that the liver is involved, and the ovaries are involved. But we really don't know.
Okay.
Exactly where you need it.
Right.
Or even in what fraction of cells you need it.
And I guess I phrase it like that because as I started learning a little bit about gene therapy, one of the things that I've learned is there are certain vectors that they need to try and use. And one of the ones that a certain company is trying to use is they're using a certain vector because it crosses the blood-brain barrier, and so it
Yes.
can be able to get into the brain, and so like that's an important thing, and that's something that you guys were going to look at for the bone marrow transplantation as well, like if it can affect in those same areas.
So, I mean, we also, as part of other projects, we do gene therapy, and we also use vectors that can cross the blood-brain barrier, and we do that intentionally because we think it's pretty important that you get some GALT activity in the brain.
So brain, liver, you mentioned ovaries. Are there other areas that are kind of focused on as probably needing to be important?
Those are the big ones that I can think of. Shauna,
Okay.
Maddie, do you have other?
I was gonna say maybe muscle only because we do have some movement disorders in our rats that we've been evaluating as well, such as like balance and stuff like that, which is kind of a cross between brain and
It's
muscle.
kind of neurological, but
Yeah.
we don't
So,
really.
so I would say muscle is another important one as well.
And when you look at muscle, because obviously your muscle, like your brain is just in your head, but muscles are basically throughout your whole body. So are you targeting certain muscles that are easier to target? Like how does that all work? Or is it just like we're going to try and get them into the muscles as well?
We try to get it into the muscle. I mean, the majority of your body is skeletal muscle, and that's primarily the muscle we look at. We have not looked too much at cardiac or smooth muscle, but those are definitely things we do want to look at. But for right now, the majority that we have looked at has just been skeletal muscle.
I didn't even know there were different types of muscle.
Oh, yes.
I just thought
Your
we all...
body's really complicated. Yep.
Yeah, it sounds like it. Okay, all right. So we've got this idea of bone marrow transplantation. Let's get back to that. Let's just start there. When you go into this study, what are some hopes of what you are, like, what are you hoping to see as you start this study?
Well, we wanted to, this study had multiple parts because, we talked before that the bone marrow donor has to be matched with the recipient. So I hoped that we could make a matched colony. We did. I hoped that we could actually achieve the technique of doing something called myeloablation. So you have to give, basically we gave some nasty chemicals to the recipient rats to wipe out their own bone marrow, so there would be space for the donor marrow to come in. The very first time we did this, we didn't want to hurt the rats, and so we gave a very small dose, and we didn't get engraftment. So we had to go back and say, okay, let me look at some literature, and we ended up doubling the dose, and that's what worked. So I hoped we would get engraftment, which eventually we did. And then I hoped that the engraftment would solve all the problems and would, fix all the metabolites, not just in the blood, but in the brain and in the liver, which are the other places we looked. And as we'll get into, the bone marrow transplant worked. And we basically, we fixed the red blood cells, but it didn't fix the liver or the brain.
Got it. And that's a super important
Yeah.
finding. So Maddie, if we can jump over to you real quick, I'm interested, what was your role in the study and how were you involved?
Yeah, so we kind of started the bone marrow project right at the same time as I was entering my junior year of college, in which I did research for credit, where Emory says that you can, instead of doing a class and learning all this stuff, let's get some actual hands-on practical experience. You have an independent project in collaboration with the laboratory, and both do the actual research on the bench and also learn how to, write up methods and abstracts and all that kind of stuff. So this was my project for my junior year class. I had done some other projects in the lab, but Judy kind of came to me with this project very early on. It's like, this can be your project for the class, and start from ground zero, designing the experiment, breeding rats, working on literature review, and figuring out, like Judy said, the dose for the busulfane to knock out the immune system. So yeah, it was nice to be able to see a project as a young researcher get built from the ground up. Versus a lot of times as scientists, we jump into a project when it's like halfway through or 3/4 of the way through, and you have to catch yourself up really quickly.
Yeah. Are there, and this is just, excuse my ignorance, but like, you know, when I work in entertainment, right, there's a producer, there's an editor, there's a director, there's a host and talent. Is there, in a research project, do you guys have titles and responsibilities and things like that?
I would say that it's not that necessarily a title. I mean, certainly Judy is the PI and Shauna is lab manager and staff scientist. And, you know, we'd have a gaggle of undergrads as well. But you quickly figure out whose roles are going to go towards what, which person's more writing focus, which person's going to be more on the bench, which person's going to be more designing the actual experiments or doing the literature review that's necessary to figure out all of the fine details and see what else is out there in the field. So it's not that you hire, you know, I'm also a theater person, so I get the analogy. It's not that you hire a producer and a costumer and a choreographer. It's just you all kind of settle into your roles based on your expertise.
Okay, cool. And so with that, then, Sean, if we can pivot to you, where did you fit into this puzzle of you know, this specific research project, and where did you best fit?
So luckily for me, during my grad school years, I actually did bone marrow transplants. I did them in a little bit different model. I did them in genetically immune compromised rats. So these were rats where certain genes were knocked out that impacted their immune systems. So I actually had to transplant them to make sure they were healthy over time. So I gained those skills in my grad school. So that was something I was able to bring into my postdoc here. And so when Judy brought up the idea, I said, hey, I have experience with isolating bone marrow and doing the transplants. This is something that's not necessarily difficult, but there's steps that you got to be careful about. And so I said we could easily do this. However, our animals are not immune compromised genetically, so we had to look into what would be the best way for us to make them that way. So we could have approached this in two ways. We could use drugs, which is what we ended up doing in this study, or we could use radiation. And at the time, we were like, let's maybe not do radiation. Let's focus more on chemicals. That's a little bit easier for us to do here in the research lab. So then, in conjunction with Maddie, we started doing lit searches and saying, okay, what drugs have people used? What outcomes did they have? How much did they use? So all three of us really worked together to kind of design a couple of different protocols that we wanted to test out. And we tested out one, as Judy mentioned, where we did a single dose of the busulfan, and that didn't work out. So Maddie and I were like, back to the literature. What else could we do to improve this? And we found some papers with mouse that said, hey, we did 2 doses, and that really seemed to work. So that helped us design the protocol. So then when we finally were ready to go, we're like, we know how to do the isolation. I know how to do the injections. So we were pretty much now set to kind of go about our experiments and things like that. So from there, we were able to do, blood draws and things like that throughout it, which were all things I developed skills in grad school. And in addition to Maddie, she learned how to actually do the injections of the busulfan, which is important because that has to be done very carefully. And so she was integral in helping me do that kind of part. She learned how to do the isolation of the bone marrow as well, because that does take a lot of work. We have multiple rats you need to isolate from. So she learned those skills as well. So everything I learned in grad school I was able to bring here, and she was able to learn from my experience as well.
That's awesome. That's just like happenstance, but so much synergy of just like, oh, my skills lend to this and your skills lend to that. And I happen to be able to, I know how to do bone marrow transplants because I've done them before in rats. And that's just like, it's really cool that all of your experiences kind of matched up that way. And you ran these experiments on what we've talked about before, which is a Galtonal rat model. And we went over this in episode 1 of the Galactosemia podcast, actually. So it's been a little bit. Can you guys just remind me and remind people why these rats are a good use case for things like studying galactosemia and what a Galtonal rat model actually is?
Sure. So the Galtonal rat that we have is an animal model of classic galactosemia, because there's classic and Duarte. So we focus on classic here. And that was actually created in collaboration with the Gertz lab at the Medical College of Wisconsin, where I actually did my PhD work.
Yeah.
And since I joined the lab here at Emory, it's been now characterized in the lab for the last several years. Now, while it does not mimic all the symptoms that patients experience, our Galton rats do show metabolic abnormalities and some phenotypes that are reminiscent of patient outcomes. And this is important because these biological and other phenotypes in Galton rats allow us to investigate different interventions such as BMT and whether they could potentially improve the outcomes of patients with CG in the future.
You mentioned that not all of the rats show the same experiences that people with classic galactosemia show and have. What are some that they do, and what are some that you have seen in the past, and some that you haven't, that you don't see in the rats?
So one of the most obvious ones that we have is cataracts. So our rats do develop cataracts of varying degrees, and that is very similar to what we see in patients. Some patients develop stronger cataracts versus others. That is one of our very obvious phenotypes. Our other ones are not as obvious. So we do see somewhat of a growth delay in our rats. They do catch up eventually, which is very similar to what you see in patients. They start off a little bit smaller, but they do eventually catch up sometimes to their counterparts. One thing we have seen in our rats that's not similar is while females have issues with their ovaries, our rats do have issues with their ovaries, but not as significant of a problem that we see in human patients. We have some issues, but not exactly the same. And then some of the other things we see are we have some motor and learning difficulties in our rats, which are kind of hard to assess and compare exactly to humans, but that does tell us that we do have maybe some learning difficulties, some motor defects that are somewhat similar to what we see in patients.
Yeah, I was gonna say one of the things that we are presented with from the very beginning are a large amount of speech and sound disorders, and that would be really hard to figure out if a rat has a speech and sound disorder.
We tried.
We don't.
You tried.
We were looking at their vocalization. We were recording their calls as pups, and that ended up being a lot more difficult than we thought it would be.
We don't work on that anymore.
Yeah.
I would imagine that would be really, really hard. We talked to Sineva Hagen-Lilovic a few episodes ago. She used to study in Dr. Kent Lai's lab, and she did a purple sweet potato study. Do you guys, just a question I had that came to me was, do you know if the rat models that you all are using is the same rat model that Dr. Lai uses in his lab?
So he uses a mouse, not a rat. And let me just explain one reason why we use rats. I mean, so rats obviously are bigger than mice, but they're not just big mice. Do
they have other differences? I don't know the difference between rats and mice.
They do, and I don't know, Shauna, do you want to...
Yeah, so, I mean, Judy had mentioned that they are large, and so that's obviously the biggest one. Rats, they're, for toxicological and pharmaceutical studies, they are much more well-studied than mice, and we have a better understanding of their CYP450 enzymes and how they metabolize drugs. And so that's the reason why you'll see a lot more drug studies done in rats compared to mice and things like that. And physiologically, rats are actually more similar to humans than they are mice. And so that's another reason Judy decided to go with rats over mice.
Yeah, one other reason that I want to stress is most of the laboratory strains of mice that people use are inbred. So what that means is that their background genetic information is very similar from mouse to mouse. But what that also means, because background genetic information can really influence the way a given mutation leads to outcomes. I mean, there are lots of examples where people have identified a given outcome in a mutant mouse, and then when they put that same mutation into a different strain of mice, they get a different phenotype. The rats are outbred, which is like people, so it means that they don't have all of their genetic information shared. So our Galtonal rats all have the same, they're all homozygous for the same Galtonal mutation, but the background genetic information is variable. So for instance, sibling rats, just like sibling people, would share half of their alleles at, basically, we call it identity by descent because they came from the same parents, whereas rats born into different litters to different parents will have little differences. And we wanted very much to make sure that we were not focusing on phenotypes that were really specific to a given genetic background, because in people, we have lots of different genetic backgrounds. We wanted to focus on outcomes that we would see regardless of genetic background.
That makes a lot of sense. And I guess my next question would probably be best suited for Dr. Lai or Bajina, who's in his program. But do you know why Dr. Lai chooses a mouse model? I assume there's probably benefits to that.
You'll have to ask him. I don't know.
Right. I will do that.
Okay.
Next time I talk to him.
Okay.
Why do you do mice? I'm sure he's got an answer. So
I would expect so.
Okay, so let's get back to this bone marrow transplant procedure. You guys are getting ready. You've kind of you've figured out the dose that is working. When it comes to the actual procedure, how do you prepare a rat? Do you have to put it under? Do you, and where were the bone marrow cells harvested from? How were they administered? Can you walk us through that procedure?
Sure. So as mentioned earlier by Judy, we actually bred a special little colony within our larger colony to have animals that had the exact same major histocompatibility genes, so that way we could limit the amount of rejection that could potentially occur in our animals. At the same time, some of these animals actually were still GALT positive and others were still GALT null, which then allowed us to essentially develop matching pairs of donor and recipients that we wanted to be able to transplant. At the same time, we actually bred in an additional gene that expresses green fluorescent protein, which is actually originally from jellyfish. We bred that into our colony so that our bone marrow donors would have cells that would fluoresce green, while the recipients would not have any green cells within them. And then this actually gave us a really easy way to follow engraftment of the donor cells using a method known as flow cytometry, where we could actually count how many green cells were in a sample of blood or bone marrow. And so for the engraftment of the donor cells to occur, as mentioned earlier, we actually needed to prepare animals with a chemotherapeutic drug. And for us, we actually used a drug called busulfan, which is one that's very commonly used in the clinic and in research. And this causes a myeloablation to occur typically before stem cell transplants in the clinic. So we mentioned we tried this protocol before with a single dose and we did not get our desired results. So Maddie and I went back through the literature and we said, hey, let's try 2 doses that are a little bit lower and see if that would help us get better outcomes. So you went ahead and then tried this 2 doses over 2 days back to back. And then what we did is the next day after their 2 doses, we collected our donor cells from siblings that had been bred with their MHC match and had the GFP. And then we went ahead and isolated those cells. So to be able to do that, we went ahead and we euthanized our animals, and then we isolated bone marrow from the large leg bones in their hind legs. And that's very typical because that is one of the sources where you can get a lot of bone marrow cells out of. And then what we did is we went through our protocol where we had done some washings of the cells, some centrifugations to remove other cell types, and then we went ahead and administered those to our recipient rats. And we did that via a tail vein injection. So we put our animals under anesthesia where they would fall asleep, and then we would clean their tails up with some betadine to make sure we had a sterile environment because, as mentioned, these are now immune-compromised rats. And we would go ahead and insert our needle into their tail vein, make sure we were in their tail vein properly, and then go ahead and inject the cells into their body. And we would hope that once in the body, they would migrate around and hopefully take up residence, let's say, in the bone marrow and potentially in other cells. But we did come across some challenges with this process, and that's typically with the isolation process itself and the injection. So we wanted to make sure we can achieve high quality donor cells. So when we isolate these cells without damaging them is very critical. So that means you can't be too rough with them. You can't mix them too hard. You want to be careful when you're centrifuging them, that you don't overspin them and things like that. And as I mentioned earlier, we did tail vein injections. Tail vein injections in themselves are actually not that difficult. It is something that, I learned over a couple of weeks and have become very proficient at. But the thing is, every animal is a little bit different. And so sometimes you have animals that have easier vein access where, like, as soon as you put that needle in, you're in it. And then sometimes we have animals where their tails are a little bit different. They have maybe their veins sitting a little bit lower inside. So sometimes it can be difficult to actually get the needle in and make sure you're in where you want to be. But we make sure that we are adequately within the tail vein prior to injecting the cells, because that's key. So having a good injection will help us have the possibility of a good engraftment.
When you've mentioned, we've talked about engraftment a few times, and it seems to be a marker for, like, you guys didn't get good engraftment on one, and then you did get good engraftment. What is engraftment? So
engraftment is essentially like the donor cells taking up residence and repopulating the recipient's cells. So
okay.
What we consider a good engraftment would be an animal that you transplanted and then you see lots of green cells. So that will tell me that those cells did get into the bone marrow. They were still healthy enough after the isolation and injection that they took up residence, and then they were able to reproduce, meaning they were able to respond to the recipient signaling, say, hey, we need to make more of these cells now. And so they were able to respond and repopulate.
Yeah, just to be clear, so the experiment for us lasted for 10 weeks, and we don't harvest bone marrow until the very end, and we euthanize the animal. So when we collect just a little bit of blood sample, basically while the rat is alive, Shauna can anesthetize them, take a little bit of blood. We're not looking at engraftment into the bone marrow. We're actually asking, have the donor cells engrafted in the bone marrow, and now are they producing mature blood cells that are in circulation? Because we're actually measuring cells from the circulation. So if we see them in the circulation, we basically know that they must have come from the bone marrow, just because
right.
That's the way, and we're not looking at the red cells, we're looking at the white blood cells. So it's a little bit of a, couple step process. And again, we're looking at blood in circulation and saying, do we see green cells in there? If we see green cells in there, we're like, yes, the donor cells engrafted.
It must have been through this process that
exactly.
we just did, which means it was a successful, at least that part of the process was successful.
Once the experiment was over and the rats were euthanized, then we did actually directly take some bone marrow and say, do we see green cells in there too? And
right.
It was what we expected, but
of
you
course.
Can't do that till the end.
Right.
So.
One of the things that I've found out through this show that's important when you do studies is biomarkers. I didn't really know what biomarkers were, but to put them, in my own experience, when we test Elliot's Gal-1P levels or his galactitol levels, if he were going through some sort of therapy or something, those would be good biomarkers to test and say, and maybe I'm wrong, I don't know, is that a skeptical face, Dr. Judy, that maybe those aren't good biomarkers?
Well, so galactose-1 phosphate in red blood cells and galactitol, which is often measured in urine, are markers of disease, which means you can tell if the person has galactosemia or doesn't have galactosemia. If we had 100 people with galactosemia, their Gal-1Ps would be higher than the 100 people without galactosemia. Okay?
Right.
But if you're asking, does the Gal-1P correlate with, are they going to have speech problems or are they going to have cognitive problems? It doesn't correlate.
Right. But in terms of if you're trying to get rid of galactosemia, it might be a good biomarker to say the galactitol levels have been severely reduced now that this person has gone through gene therapy and doesn't have galactosemia anymore, in a hypothetical world where that exists.
So following these galactose metabolites tells you if you have, in the instance of gene therapy, if you have restored the ability of the cells to metabolize galactose, because they're not building up these metabolites anymore, so you must have restored the ability to metabolize galactose. And we feel in our gut that must be a good thing because we think that it is impaired galactose metabolism that somehow, connect the dots, leads to cognitive problems or speech problems. We don't really understand how to connect all those dots. So these metabolites are markers of how well is the body metabolizing galactose.
Right. And so those are things that you're viewing, i.e. biomarkers. Is that right? That you're able to use in a trial or a test or whatever, a research that you're doing. You're looking for biomarkers that you can look at and say, like, is this doing what I intend for it to be doing?
Well, so you have to be really careful with the word biomarker because, yes, it's something we can follow. And in some conditions, for instance, in phenylketonuria, they know that it's the phenylalanine level that is toxic to the brain. And so if you lower the phenylalanine level by some intervention, you really know phenylalanine is the bad guy. And if we're lowering it, that's going to be better for the brain. So for galactosemia, it is my opinion that we don't really know who the bad guy is. So we know that Gal1P goes up, but it doesn't really correlate with outcome. And we know that galactitol goes up, but it doesn't really correlate with outcome. Maybe it's a mixture. Maybe it's a combination. Maybe it's different things in different tissues. We don't know. So yes, being able to say, I made these metabolites look more normal than they did before is certainly something that would make us happy. But I don't think we could say, therefore, we've solved, the cognitive problem or the speech problem or whatever, because I think in phenylketonuria, the phenylalanine correlates with these outcomes. In galactosemia, it doesn't correlate. So there's a little bit of a uncertainty there.
Yeah, I think that. Oh, sorry.
Go ahead. No, go ahead, Maddie.
No, I think as scientists, and the way I think Judy is alluding to is we try at every possible place to not say A equals B, therefore B equals C, therefore solution, right? Like we're trying we're not absolute people unless we know 1,000,000% that that's the absolute. So in terms of the question of biomarkers for galactosemia, we know that having a deficiency in the GALT enzyme increases your galactose, galactitol, and Gal1P metabolite levels. In the bone marrow transplant, if we see those decreased, we think that might be a correlation of increased GALT activity, but we're never going to say that 100% that's fixing your galactosemia.
Right. And I guess that makes sense. It's hard to wrestle with the fact that there is not that correlation, but also that your kind of logical mind says if I have a very reduced galactitol level and a very reduced Gal1P level to a place where a person with GALT enzyme activity it's at that same level, then I have essentially given that GALT enzyme activity, i.e. cured the galactosemia, and therefore probably also cured the speech and sound disorders that would come along with it, or those types of things. So
it's the probably, and we're sure of, and we theorize that scientists will use, because if you see a case where the data doesn't agree with that, that's also important, but it's never going to be true 100% of the time or in every patient sample or something like that. The probablys and the maybes and most likelies and we theorizes, those are important questions and they're important conclusions. But yes, that's the whole issue with the whole biomarker turn.
Right. And so I guess going back to all the way to what I was kind of initially going to ask was, like, when you guys looked at this experiment, were you using biomarkers that you were measuring, and if so, what were they, and how did you measure them?
Yeah. So we measured two different types of quantifying markers or biomarkers. We mentioned the metabolites. So we measured the amount of metabolites, galactose, galactitol, and GAL1P in different tissues. So galactose and galactitol we measured in the liver, brain, and the blood, so the red blood cells in the plasma. And then GAL1P is strictly an intracellular metabolite, so that's not found in the plasma. So we just measured that in the RBCs in the liver and the brain.
All right, hold on.
So I'm looking at those.
Time out.
Sorry, a lot of terms. Yes.
No, all right. Break down, because I understood the galactitol, and then you got to GAL1P, and you said a bunch of words I didn't know about how
Yeah,
it's a...
sorry about that. So galactitol is what we call an intracellular metabolite, where you have intracellular and extracellular, where some metabolites are made in the cell and they stay in the cell. That's what intracellular means. Whereas other metabolites are made in one cell, and then if my neighbor cell needs that metabolite, I can share. So that's extracellular. They can move in between.
So what Maddie is getting at is that some molecules are able to cross the membrane. So cells are surrounded by a plasma membrane, and there are some molecules that can get across that, either because just chemically they can get across it, or because there are specific channels that let them out, for instance. So galactose and galactitol can get out of a cell. GAL1P can't. So GAL1P is made inside the cell, and that is where it stays. And so red blood cells are, cells, and so they can have galactose in them, galactitol in them, and galactose 1-phosphate in them. Same for liver, same for brain. Plasma is an extracellular fluid. So plasma is not a bunch of cells, it's fluid that is kind of bathing cells. And so typically, even if a person has galactosemia and is drinking milk, you won't see galactose 1-phosphate in the plasma unless you've this sample is old and it's had hemolysis or something, and the red cells have busted and the GAL1P has come out. So
is that why you typically test galactitol levels through urine and GAL1P levels through blood?
Yes. And you could, we measure galactitol in the plasma or in the red cells, or because we can measure it in cells or not in cells. But because the kidney normally filters out galactitol, it concentrates in the urine. So it is basically an easier target, so it's easier to find in the urine. So that's why they measure it there.
How often, this is just a biology question, I think that more people than me probably already know. How often do red blood cells regenerate?
So in humans, red cells typically last about 120 days.
Okay.
And now there are, if you're iron deficient, or, you've lost a lot of blood through something, you may be producing them faster. So there are all kinds of situations that can influence how quickly you make them, or how long they last. But 120 days is kind of what the textbook would tell you.
Got it. And I asked that question because I believe, it's my opinion, we're getting closer and closer and closer to gene therapy in galactosemia. And one of the questions that came up at a thing I was at was, galactitol can leave the body through urine, but GAL1P, what we're saying is it stays in that red blood cell. It's stuck.
Unless it gets metabolized. Right.
Right. So if you are building up this kind of GAL1P in your body, it's not stuck there forever, because your red blood cells are regenerating every 120 days.
So the red cells are turning over. That is correct. GAL1P, even without GALT, GAL1P can be metabolized, just not very well. Okay, so there's something called a bypass pathway, where there's another enzyme that is abbreviated UGP, and it doesn't normally work on galactose 1-phosphate. It normally works on glucose 1-phosphate. It's the enzyme that normally takes something called UTP and glucose 1-phosphate and makes UDP glucose. And UDP glucose is what your body uses to make glycogen. So your liver has a ton of UGP because it needs to make glycogen. All the other cells in your body need to make glycogen too, just not as much as the liver. If galactose 1-phosphate builds up to high enough levels, that UGP enzyme will work with it. It doesn't like to work with it. It's not very good at working with it, but it can. And so it's not true that galactose 1-phosphate has absolutely nowhere to go. It's just going to go slowly. Okay.
Right. And again, so like if we, let's say we live in a world where we're doing gene therapy and we activate the GALT enzyme, then eventually you're regenerated to where your levels of galactitol and Gal1P are that of a person that has, it's not like it just builds up and is going to be forever high levels. Like you'll eventually get back to a normal point.
We do gene therapy in our rats and we lower all the metabolite levels, mostly not exactly to normal, but pretty darn close.
That's amazing. It's so hopeful for
families. Yes.
It's honestly,
we're very excited. Yes.
Okay, so one of the things that you mentioned in the paper that I thought was super interesting was that there was a human subject that has galactosemia who was given bone marrow transplant, but as a treatment for leukemia. Can you tell us about that?
Sure. So let me just say that I didn't know about that young man when we did this study. Okay, we did this rat study a while ago. And after, we had already basically done the study, I got an email from a young man who has galactosemia and also came down with leukemia. So, people with galactosemia can get all of the same other things that can get the rest of us. It's not protective. And this young man had come down with leukemia, and so he had the standard of care treatment, which was a bone marrow transplant. And the bone marrow donor didn't have galactosemia, so those cells
would be crazy if they did.
Yeah, it would have been very rare. And after the bone marrow transplant was successful, his doctor measured the GALT activity in his red blood cells, which were now not really his red blood cells. They were from the donor marrow, and the GALT activity was normal. And so he contacted me and said, does this mean I don't have galactosemia anymore? And because we had done this rat study, I had to say, not really.
Right.
Your red blood cells don't have galactosemia anymore, but the rest of your body still does, or probably still does.
Meaning the cells in his liver, the cells in his brain.
So on.
Interesting.
Yeah.
And what effects? So that was the result of what the study. I don't think we've even said that yet. The result of your study was you found that the red blood cells restored activity. The other areas you looked at did not.
Correct. And so the red cell Gal1P was normalized, but when we looked in liver and brain, it was not normalized at all.
And that in your, I guess, in you guys's what you were looking for, does that mean that, like, and I don't know if you even put it this way, but the experiment was a failure, how do you quantify that? Okay,
I would say that the experiment was a success in that we got engraftment, and we were, so we got lots of green GALT plus cells, and they, we restored GALT to the red blood cells. We restored, it was functional GALT because the galactose 1-phosphate was normalized, but it was certainly not the result that I was hoping for. I mean, the result an experiment tests a hypothesis. If the experiment is a success, sometimes it validates your hypothesis, and sometimes it says that your hypothesis was wrong. So this experiment was a success, but what it said was that my wishful thinking hypothesis was not correct, or at least was not correct in the way that we did this experiment. So, for instance, we used adolescent rats. It's possible if we had done it in newborns, maybe it would have worked better. We used busulfan to myeloablate the rats. There's a lot of research being done to find new and less toxic and more effective ways to achieve myeloablation in people who need bone marrow transplant. And it is possible that some of those new methods may give bone marrow-derived cells better access to the brain or the liver. So I don't want to say no way, no how ever is bone marrow transplant going to work, because I don't know that. What I can say is the way we did it, the result we got, was not something that would make me say, hey, this is a great thing for galactosemia. But it is very possible that someday down the road, with these new, better myeloablation, and the other thing I should add is that, we had to engineer, sort of a little colony of rats that were all, major histocompatibility identical with each other, so they, were perfect matches, kind of. For people, the way I envisioned this, if this had worked the way we wanted it to, so in adults, blood is the stem cells that give rise to blood come from the bone marrow, but in utero, that's not the case. And when a baby is born, the cord blood is full of stem cells. It is like magic, okay? And so what I was hoping was that if they had saved the cord blood, you can actually isolate these stem cells from the cord blood, and you put them in a dish and you do gene therapy on them in the dish. So it's the child's own cells that you are giving GALT in the dish, and you can test it and make sure it worked. And now they've got GALT, it's functional. And then you can give those very same manipulated, but the child's own cells that you didn't collect from their bone, you collected it from their cord blood when they were born. So you would have thrown it away otherwise, that you could give that back. And so they do that for children with other types of conditions where it's called ex vivo. So you do gene therapy outside the body. The cells are in a dish. You do the gene therapy, and now you give those, call it corrected cells, back to the child. They are a perfect match because they are the child's own cells.
Right.
You don't need to, have an identical twin or breed a colony or something. So that is what I was hoping we could do if this worked in the rats in a way that was protective of the rest of the body. I was hoping that this could be done basically in very young babies using their own cord blood cells.
And in what your study leads you to believe is that if you were to do that, your red blood cells would gain GALT enzyme activity, but the cells in where we believe you need them in the brain and the liver, etc., would not. Not
using the methodology that we used.
Tested.
Again, I have served on people's dissertation committees who are doing some of this stuff, so I know that there are amazing advances happening in myeloablation, and some of them are using special kinds of antibodies and stuff that are not just toxic chemicals, which is what busulfan is. And so it is very possible that as these new approaches get better, they may not only achieve making space in the bone marrow, but improving permeability of the blood-brain barrier, which is, and basically helping the white blood cells that come from the bone marrow be able to find their way into these other tissues. So I don't want to say that'll never happen. But
We're still holding out hope.
I'm holding out hope, but I can say the method we used was not something I would recommend for a child. But down the road, there may be improvements in the methods that would make this possible.
Yeah, and Maddie and Shauna, as we wrap up, I'm curious from your point of view, what you've learned running this study and what you learned personally about galactosemia and translational research just in general. Either one of you, how do you feel?
Shauna, you're muted.
Oh, sorry, I muted again. Gotcha.
Okay, great. So obviously working with an untested protocol always brings with it some hiccups along the way, mainly because you don't know what's going to happen. We got lucky that all of our animals essentially survived the treatment and the transplant, but then we had some issues post-transplant that we did lose a couple.
We lost 2 out of 7.
Yeah. Well, we were encouraged because we saw our animals lose weight, which is what we expected to see, and then we saw them gain the weight back after the transplant. So that was a sign that, like, okay, this protocol seems to be working out well. And then obviously working with immune-compromised animals, there's always some drawbacks because you have to just handle them much more carefully. Usually they require some kind of special antibiotic water to prevent them from getting sick while they're waiting for the transplant to engraft over the next couple of weeks. And then when you're performing multiple blood draws, that could also lead to challenges when you're essentially going up and down the tail. You could be leaving scar tissue, and that could always make it difficult to get your samples out of the animal. But these experiments showed me that we need to continue working on these new approaches for galactosemia treatment. As we saw with our work, bone marrow transplant may not be a great option at this time, but at least it could be in the future if some of these new therapies are more effective than what we were looking for. And when it comes to translational research, which has always been a passion of mine, my PhD is in physiology and translational science. And so I'm really excited to continue working on bringing a lot of our science from the bench to the bedside to help patients and their families live their best lives.
And Maddie?
Yeah, no, I agree with Shauna with that from a translational research perspective, perhaps this method, bone marrow transplants to treat galactosemia, may not be the absolute key, but translational research to me is about finding different solutions and trying different approaches and trying different ways to get what we know about the basic research side of galactosemia to the bedside of these patients and improving their quality of life and disease outcomes. So, like I said, I was an undergrad at this time, so all of this was new to me, learning it for the first time. But it was a good lab to learn not only science, but translational research and seeing how my work and the work of the lab, day in, day out with the rats, how that could actually have a beneficial outcome towards these patients' quality of life.
Yeah, you're so right. And it's just like from the patient side of things, I often talk to people like you guys and Dr. Lai or Dr. Barry or, Dr. Estella Rubio Gozalbo, who's like, you guys are doing the translational research that could possibly affect my son's life. And so that's just kind of like, I always want to say thank you because the day-to-day work that you guys do in studies like this, it doesn't just live like on your workbench or in these labs that you're doing it. It has the real possibility, and some of them we're seeing are coming out even more and more now of like affecting real lives, not just in galactosemia, but other diseases. And for me specifically, I had, obviously care a lot about the galactosemia part of it. And so there is that real world effect that you guys are having. And Dr. Judy, if you could speak a little bit to, how studies like this, whether they have good outcomes or outcomes that you wish were better, but how all of these studies fit into your lab's broader mission.
Sure. Thanks for asking that. So we are a research lab at a university, so we have really three missions, I would say. So one of them is to learn as much as we can about, learn from people who have galactosemia, learn about the experiences so that we can understand, basically what are the patterns, what should people expect, are there any things that can be used to predict, who's going to have problems and how we can best help them. And so that's the work that we do with our human subjects study. And I'll just say that a lot of what we find, I think, will also help to inform the design of clinical trials. So it's important not just because inquiring minds want to know, but it's actually, it helps with the accuracy of prognosis. It helps with the design of trials. Our second mission is really from the wet lab work, the rat work that we do. And I'll say, when I first started at Emory, I first developed a yeast model and did a lot of work with it. And then we realized we needed multicellular. And so we created a fly model and we did a lot of work with that. And then we were at the point where we were, getting galled back. And I was like, okay, we need something bigger and closer to a person than a fly. and so that's when we decided it's time to make a rat. And so, we're doing gene therapy testing in our lab. Some of it is with vectors that we designed. Some of it we're testing things in collaboration with companies. So, we're trying to learn from the people who are here now. We're trying to come up with better interventions or help to test and be part of that so that hopefully, tomorrow we'll have something better. And then the third mission really is to help train the next generation of scientists and doctors. And so you've got, two great examples here. But, each year I'm really privileged to get to mentor, usually 5 to maybe 8 or so undergrad. Sometimes we have 10, which is kind of a lot. And Shauna is definitely my partner in crime on this. And so, we have mostly undergrads, but we also have some students from, genetic counseling or from School of Public Health. And most of the undergrads go on to either medical school or graduate school or MD, PhD. We have one who's in genetic counseling training now. I always tell them,
or the NIH.
Or the NIH, yeah. I mean, they should follow their dream, and whatever they want to do, how can I help them get there? And so, this study was, one small part of, we always have a lot of projects going on. Some of them are wet lab, pipetting stuff or working with rats in the lab. Some of them are working with patient information or, doing stuff on computers or talking with people. We would call that more dry lab. And then we're always trying to put these things together and say, is what we learned from the rats useful for the people? What did we learn from people? Gee, should we test that in the rats? A study that we're trying to finish up now that Maddie was very involved in and Shauna is also extremely involved in is something you can't do in people, which is ask what are the prenatal consequences of GALT deficiency. So that's something that I hope will be coming out hopefully in the coming months. That's been a project that has been that was a very heavy lift. I'll just say very heavy lift, but it is very close to done now. So anyway, I really feel extremely privileged to get to do what I do. I never ask myself why am I getting out of bed in the morning. So.
Yeah, I love that. I always like when we talk about experiments to give a shout out to funding because that's so important. How is this funded?
Sure. So the bone marrow transplant project received some funding from the Galactosemia Foundation, and so I'm very grateful for that. And it also was dependent on previous funding that we had gotten from NIH. So we had a grant from NIH basically to develop the rat model and characterize it. And without that, there's no way we could have done the bone marrow transplant project. And so anyway, so I would say that both of those sources were key.
Another example of why keeping funding from the NIH to college and universities doing research is so important.
Yes.
Just want to,
Indeed.
just put that out there for everyone to hear and make sure everyone knows that is still super important, that we allow
Yeah.
the NIH to fund projects like these.
I always tell people that, there's a story, I think it's not from the Bible, but it's from something like the Bible, of an old man was seen planting fig saplings, and the person said, old man, you're not going to live to eat those figs. And he said, no, but my children will. And funding basic research is kind of like planting fig saplings. There is a 50 plus year track record proving, that basic research eventually saves lives. And right now, a lot of people, get Alzheimer's or get cancer or galactosemia. There are conditions that we don't yet know how to cure. And I would very much like, by the time my children are old, or if I ever get grandchildren, I would like for there to be a cure for cancer for the next generation. It may not be good enough for my generation, so anybody who takes a statin drug, the drug was actually developed by a company, but how did they know which enzyme they needed to inhibit? That was done in a university. So there's just, almost any major advance that has touched healthcare, or like GPS. Do you use GPS? Guess where that came from? It's not only the NIH budget that's at risk, NSF, which funds a lot of the sort of physical sciences, and our lives are better because our parents paid into these systems for public funding for science. And I hope we care enough about our children and grandchildren to keep paying into it so that their lives can be better than ours. But I'm scared. It's a tough time.
That's exactly right. And you're more than welcome to stand on that soapbox on this podcast at any point in time. We've made it very clear where we stand and how important NIH funding is, and how dangerous what the current administration is trying to do is. And I'll just leave it at that. You mentioned some other research projects. I want to ask you really quick about what other stuff do you have going on at the lab right now that maybe is interesting?
Sure. Well, there's a lot, so I'll try to give you just some general highlights. So in terms of human subject studies, we've done a lot of work asking about, like, at what age, we call it penetrance and expressivity. So a couple of years ago, we did studies and showed that it's not progressive. So it's not like it's going to get worse over time. More recently, we did a study looking at penetrance and expressivity, which means, like, some people have a speech problem and some don't. Okay, so speech issues are not fully penetrant in galactosemia because some people have them and some people don't. Expressivity means if I took 100 people with galactosemia who all have speech problems, they wouldn't all have exactly the same severity of speech problems. So expressivity is like, okay, you have the complication, but how bad is it? Mm-hmm. Okay, and so we knew from many studies by our group and many others over many decades that, roughly what the prevalence is of complications. But people had not done studies looking at by what age are 90% of kids who are eventually going to be diagnosed with a speech problem. So if my kid is 6 and they don't have a speech problem, do I have to hold my breath because tomorrow they might have a speech problem? I can tell you no, it's fine. The median age at diagnosis for speech is 2. And by age 4, 90% of them have been diagnosed. For cognitive, I think the median was 5 and 90th percentile was 8. And for motor, median was 3 and 90th percentile was 8. So this is all very useful information, especially, for families, for healthcare providers, but also if you're trying to design a clinical trial, at what age do we need to bring people in? And then how long do we need to follow them? And one of the things we're actually gearing up to try to do now, but I just submitted a grant to NIH this past Monday, and we'll see if it gets funded. So we've pretty much shown, and others have shown this too, that subtle differences in diet really don't correlate with outcome. And the quote unquote biomarkers, the metabolites, really don't correlate with outcome. So how the heck are we supposed to predict, if I have, 80 babies are born each year in the United States who have classic galactosemia, and we know that by the time they're in elementary school, at least half of them are going to have developed some kind of developmental complications. But looking at them as babies, they all are beautiful babies, and I couldn't tell you which ones are going to have problems and which ones aren't going to have problems. And so we have strong reason to believe that we talked about background genetics. We think that there are genetic modifiers. And so I actually just submitted a grant to try to identify them, because if we could identify them, then you could, in a little baby, have a much more accurate, I'll call it sort of personalized prediction, and not just say, well, there's a 50-50 chance that you're going to have this problem. We could make that much more accurate. If you're designing a clinical trial, you could know that, the kid who's not going to have the problems anyway probably shouldn't be in your clinical trial. They don't need it. And once approved treatments are available, the parents and the doctors are trying to do risk-benefit analysis. No treatment is perfectly safe. Is it in the best interest of this child to give them,
Yep.
fill
It's
in
a
the
very
blank?
real thing that, we're going through it in terms of, like, let's say clinical trials for gene therapy get underway, and it's like, do you want to be a part of something like that? We don't really know for Elliot how bad things are going to be. He's meeting
Exactly.
milestone. It's like you just don't know.
Right,
So
but-
it's such a hard thing to grapple with.
But if we can identify the genetic modifiers,
Right.
then, they could basically get
Yeah.
some saliva, do a DNA test, and
Right.
say, your child has, instead of a 50% chance, push that,
Yep.
up or down.
Up or down.
That's going to help. So, so that's basically what we're doing with the human work. We're also doing a deeper dive into ovarian function and into neurological outcomes, tremors and seizures and stuff. And in terms of the rats, we're continuing with our own gene therapy work, and some of that is, I'll call it more traditional gene therapy, and some of it is testing an idea for doing gene therapy that would not only teach cells how to make GALT, but teach them how to share it with their neighbors, which would be really nice because gene therapy never hits every cell. So that is very exploratory work right now, and and we'll see how far we can push that. And then we're also doing some work with some companies. Basically, they have things, and we use our rats to test them. And I probably shouldn't say any more about it.
We'll leave it at that.
Yeah.
We're going to be doing an episode soon with a couple of companies that are working on gene therapy for galactosemia.
Good.
So we'll dive in there. How long before I can do an episode with you on what you were just talking about, which is the gene therapy giving GALT to its neighbors experiment thing that you're doing? Like, how far are you in that, and can we talk about that?
Why don't we wait until we publish our first paper on it?
Okay.
And then, okay. So.
All right. You let me know.
Okay.
Well, thank you, all three of you, for joining the podcast, for taking the time. I really appreciate it. I know the community really appreciates it. And it was really, really great to have your insight. And thank you for doing the work on the experiment that you did. Every little piece really helps. So we appreciate it. And thank you guys so much for giving your time to us.
Well, thank you, Ryan. This has really been a pleasure.
Of course.
Absolutely.
So
Thank you.
Okay. Thanks, guys.